சொல்லுங்க. அவங்க இப்ப இன்ட்ரோ듀ce பண்ண போறாங்கப்பா. பண்ணுங்க. இது பண்ணுங்க. அவங்க பேசுறது கேட்கணும் நமக்கு.
A very happy morning to one and all. Myself, Dr. S. Arif Nisha, Assistant Professor, Department of Biotechnology, Srimad Andavan Arts and Science College, Trichy. It's my immense pleasure to welcome you all for this second day of two-day Science Academy's lecture workshop on recent trends in microbiology and biotechnology. First and foremost, I would like to extend my warm welcome to all the resource persons, the convener of the workshop, Dr. Tapan Kumar Adya, Professor, School of Biotechnology, KIT University, Bhubaneswar, and Dr. DJ Bagiraj, NASI Honorary Scientist and Chairman, and Dr. N. Tajuddin, Professor and Chair, and Coordinator, School of Life Sciences, Bharatdasan University, Trichy. I would also like to welcome all the staff members, HODs, Deans of our college, and the student participants from various institutions. I wholeheartedly welcome you all. Let's start the program with the prayer.
वन्दामहे श्रीमदाण्डवन् पादुकाङ्कम् श्रीरङ्गक्षेत्रपवित्रोज्ज्वलपारिजातम् यत्र मानवाः सर्वे सन्मार्गदर्शिताः नित्यम् पठन्ति ते विज्ञानकलाभाषाः पाठातिरिक्तविषयेऽपि प्रवृत्तास्ते सुशीलगुणावपि अनुचरन्ति नित्यम् नानाकलाक्रीडेष्वपि सुशोभिताः सर्वत्र सर्वदा तेयात्मप्रभावैः ओषधिवने स्थापित प्रयोगशालासु सम्यक् प्रकाशयन्ति ते स्वप्रभावम् अत्रैवाधीतयन्त्रविद्यासु निपुणास्ते विदेशेऽपि उज्ज्वलयन्ति शालाप्रभावम्.
So it's time to start the technical session. The speaker of the technical session 5 is Dr. N. Tajuddin, Professor, Department of Microbiology, School of Life Sciences, Bharatdasan University, Trichy. He has 28 years of teaching and 32 years of research experience. His area of specialization is cyanobacteria and microalgae taxonomy and biotechnology. To his credit, he had published 342 publications, including 27 books, book chapters, and 3 papers in Nature Group journals. He had officially visited 12 countries and published 9 books. He had received several extramural funded projects worth 5.27 crores. He had guided 33 PhD scholars and 31 MPhil students. He is also serving as principal investigator, National Repository for Freshwater Microalgae and Cyanobacteria. He is the program coordinator for MSc Life Sciences, which is an integrated program, and he is the coordinator for DST PERS program. He is a member secretary in Institutional Biosafety Committee and Institutional Ethical Committee. He is editor-in-chief, Bharatdasan Journal of Science and Technology. And he had visited several countries in order to attend the conferences and symposia, and he had delivered many guest lectures, and he had been resource person for many international conferences. We are very fortunate, sir, to have you as a resource person of the speaker of these today's sessions. So I welcome you, sir, for the technical session.
Thank you, Dr. Arif Nisha, for your nice introduction, and I thank Dr. Tapan Kumar Adya and Dr. Bakiraj sir for this excellent opportunity to share my experience on taxonomy as well as biotechnology of microalgae and cyanobacteria. So now I straight away go to my slides.
Yeah, you are already identified as host, so you can upload it. Yeah, it has come. It has come. Go to slideshow mode.
So is it clear now?
Yes, sir.
Excellent, excellent.
Okay. So today morning's topic is on molecular taxonomy and DNA barcoding of microbes. This is really need of the hour. If you want to develop any kind of biotechnology, the basic and fundamental requirement for initiating any kind of technology from any kind of organism. The taxonomy is very important. Without taxonomy, you can't identify the biodiversity. As you know, we are one among the 12 mega biodiversity countries of the world. The diversity of microbes are very vast, but because of the absence of the taxonomist in terms of identification of organism, we know less than 20% of the microbes existing around us. And coming to the taxonomy, it is a ever-changing, controversial, and exciting field of biology, because each and every decade the taxonomist view are different. The identification is different. They justify their own identification. That way, there are some slight changes in grouping the organisms. For example, previously cyanobacteria, it is called it as a blue-green algae, and because of the new techniques, including the TEM and other sophisticated instruments, the so-called blue-green algae is now called it as a cyanobacteria because it is a prokaryotic nature. Likewise, there are slight deviations are there, so we have to adjust that one. The taxonomy necessitates extensive knowledge of the techniques of data acquisition and very latest analytical procedures. So the time to time the changes are there, sophisticated instrument techniques. Even nowadays we are using the confocal microscopy for taking the micro photographs, SEM, TEM, and so on. Now the taxonomists have now become endangered species category. Just now I discussed with Dr. Tapan Kumar sir. Because all the youngsters, including me, when I joined as a research scholar, my boss, Dr. G. Subramaniam, gave the taxonomy of cyanobacteria. At the time, I was totally hesitated. So other two colleagues are working with biochemistry and molecular biology. So why should I go for taxonomy? I said no. Then he said, no. Nowadays, taxonomists are just, I said, endangered species category. If you are working on the taxonomies, you are the only person available in our country. Likewise, he convinced me, and I wholeheartedly I did the taxonomy. And now people calling me as a cyanobacterial taxonomist. So it is a great honor to work on taxonomy of the various kinds of microbes. And what are the tools and techniques for molecular taxonomy? There are several tools are there. For example, if you see this slide, people normally using 16S rRNA for identification of all, not only cyanobacteria, all kind of microbes. And in rRNA, there are two different types. In case of eukaryotes, there is a 18S rRNA, and prokaryotes 16S rRNA is there, in which there are two subunits in 70S and 50S and so on. We can utilize this rRNA for the identification. Other fingerprinting methods are there, but these fingerprinting methods are useful for only differentiating the strain level, not the identification level. So here the prokaryotes, we are using the 16S rRNA. Eukaryotes, we are using the 18S rRNA for the identification system and molecular phylogeny. Everything we can do it because largest database is available. So we can utilize the database by using the bioinformatics tools. We can make the identification and phylogeny of these microbes. And in addition to this, there are other tools like ITS sequences and then chloroplast, for example, RBCL genes, then RAPD, STRR, LTRR, RFLP, then REP-PCR, then ERIC sequences. These are all the different fingerprinting techniques useful for differentiating to the very closely related species, not for the identification level. And these are all the taxonomy methods. We are following the conventional method is morphological taxonomy. This is base for almost all the taxonomy, because on the basis of morphological taxonomy, we can make the phylogenetic tree. If you match the morphological based phylogenetic tree and the 16S RNA based phylogenetic tree, both are almost similar. Such a very excellent characteristic features on morphological taxonomy or morphological features. So we can, the first step and the basic thing is morphological taxonomy. You do identify the organism using the morphology in terms of microalgae and cyanobacteria. But whereas in the case of bacteria, we have to necessarily go for the biochemical characteristic feature for the identification, because the morphological diversity in bacteria is very, very less. Only 7 6 or 7 types like Bacillus, Vibrio, Coccus, then Spirillum, then Filamentous, and pleomorphic, like etc. So based on this, we cannot identify up to the morphological techniques. So we have to necessarily use the biochemical features for identification. And then coming to the molecular taxonomy, people using DNA-based composition, DNA hybridization, RFLP, RAPD, as I said, STR, LTR, pulse field electrophoresis, 16S rRNA, and the plasmid, and so on. These are all the different tools people using for taxonomy. In coming to the biochemical taxonomy, the biochemistry is not constant. So we cannot use this for identification system, because if you change the media composition and condition, if you change the pH, if you change substrate, you cannot get the same type of protein profile or fatty acid profile and so on. Likewise, all the morphological, the biochemical markers such as fatty acid fingerprinting, people studied the various aspects. For example, free-living Anabina and free Nostoc differ in symbiotic strains. So it is not equal. Even though it is the same species, because of free-living and symbiotic, the molecular genetic makeup is different. Likewise, polyamines, the other biochemical features like enzymology, regulatory patterns, and amino acid pathways, all are different. It is based on, if you change the pH, everything will be changed. Likewise, the protein isozyme patterns, these are all used as biochemical markers. But if you want to generate a data bank for particular species, we can use these biochemical techniques, biochemical tools for developing the data bank for particular species isolated. But it is not really useful for the identification system. Then again, phycobiliproteins, DNA-based composition, restrict RFLP. As I said, these are all different tools now people trying. And coming to the GC percentage, if you see this slide, the GC percentage of the organism is not unique. Everything is overlapping. So one GC percentage of one organism is overlapping with other. So we cannot use this. If you see the Burgess manual, GC percentage is one component in the identification system. But really, if you see this slide, you understand that there is no uniqueness in GC percentage for any group. All are overlapping. So that's why this is not really useful for the identification system. Coming to cyanobacteria, people studied the GC percentage of different genera. For example, the Xenococcus, it is a unicellular colonial form. Arthospira, it is a filamentous spirally twisted form. And Cytonema, it is a false branched heterocystus form. Both are, all are morphologically very different, but the GC percentage is similar. So based on this, as I said, we cannot use this as a marker for identification. I'm coming to the rRNA, 16S or 18S rRNA. It is present in 70S ribosome in the case of prokaryotes and 80S ribosome in the case of the eukaryotes. If you see the 70S rRNA, 70S ribosomes in the prokaryotes, there are two subunits in which the larger subunit, 23S rRNA and the 5S rRNA is there, and the 16S rRNA in the smaller subunit, in addition to several different types of proteins also present in this ribosome. Similarly, in 80S ribosomes, there are two subunits like 60S and 40S. In 80S, there are 28S, 5.8S, and 5S ribosome, and 18S ribosomes rRNA in the small subunit. So this particularly the 16S and 18S rRNAs are useful for the identification and making the phylogenetic tree. Even if you see this slide, for example, in E. coli, this is just an example. The cellular components, among the cellular components, rRNA is a higher percentage. It is a 21%, and the number is nearly 160,000 rRNA molecules are there. So we can usually utilize this rRNA for the taxonomic purpose. So why is 16S rRNA in the case of microbes, prokaryotes? Why 16S rRNA is very important? If you see this, it has the regions of nearly universal sequence conservation. So if you see particular region, for it is conserved for genera, some particular region conserved for species, and some region conserved for the order and family, like this. There is a universal conservation sequences are there. And it is very easily alignment because of the length is very convenient, statistically convenient, and we can use this 16S when compared to the 18S, 23S. It is very long molecule than the 16S rRNA. It has nearly 2,500 nucleotides. Here, the 16S rRNA is only 1,600 nucleotides length. It is statistically very valid to make the phylogenetic and statistical comparison also. And it is reasonably large and informative. So it has the conserved sequences, and in addition to this, non-conserved sequences, that is variable and hypervariable regions. This region is very important to differentiate the closely related species or the same species of different isolates. By using this variation in particular region, useful for the strain level, identification up to the strain level. And this particular sequences are useful for the barcoding also. And in addition, largest database, that is RDP database, is freely available. Anyone can easily access and compare their sequences with this database, and lakhs of sequences available in the RDP database. And in no time, the system will match our sequences, and it will deliver. Our sequences are 100% related to this particular species, 98% related to particular species. Likewise, we can get these results. And this is the secondary structure of 16S rRNA gene of E. coli. If you see this, there are darker region and red color and different shades. The darker region represents the highly conserved region, and reddish and orange color, yellowish regions usually are considered as a variable region. So to differentiate the closely related strains. And these are all the different methods of DNA fingerprinting. So first basic requirement is the DNA isolation. So we need the DNA. So you can ask the question. So I'm talking about the RNA. So why we have to isolate the DNA? Because the isolation of RNA is a very tedious process. So we have to use different centrifuge, particularly it requires sometimes ultracentrifuge also needed to isolate the rRNA. And the half-life of RNA is very less, and it is very difficult to maintain RNA's free atmosphere. So it is easily degraded. Whereas if you are isolating the DNA, it is a very sturdy molecule. We are amplifying the 16S rRNA coding regions in the DNA. So we can easily get the primers and then amplify it. It is a sturdy molecule because it is a double stranded molecule. It is very sturdy. And the second step is, once we isolated the DNA, so we have to go for the PCR amplification using the 16S rRNA primers. So it will go and match only in the particular region of the DNA, and we can get the amplified products. So if you want the partial sequencing of 16S rRNA, to isolate the DNA, amplify the 16S rRNA genes and from the DNA, and we can send it to sequencing facility, and we get the partial sequencing. So that will serve for our phylogenetic analysis. But if you want to have all these 1,600 nucleotides for your phylogenetic analysis or identification, you have to go for the other steps like cloning. So you clone this amplified product with some vectors, and once amplified, you have to transfer the amplified, the cloned genes, the PCR with the vector, in the process of transformation to the E. coli cells, competent E. coli cells, and then you generate the multiple copies of the cells in the culture media. And then once you are getting the standardization phase, you isolate the plasmids, and the plasmids, you go, isolate the plasmids, and then go for the sequencing, and you get the, there are sequencing facilities are available, and you get the sequence and match the sequence using so many softwares are available, and make the phylogenetic tree. This is the overall methods, steps for making the DNA fingerprinting and the molecular identification. So I started doing the phylogeny, molecular phylogeny of symbiotic cyanobacteria using the cycuts. In cycuts, there are different group of cycas plants are there, the different families, Icadaceae, Stangeriaceae, and Zamiaceae, and we collect all the root nodules, not root nodules, it is coralloid roots. The specialized roots of the cycas plant is called coralloid roots. If you take the cross section, you can find the cyanobacterial or blue-green algal zone. So far, people reported only one species, that is Nostoc cycadiae, is reported in the cycas plant. But in our molecular studies and also the conventional morphological studies, we proved that there is no single Nostoc cycadiae. So we have more than 19 different strains and some new species also present in the coralloid roots of the cycas. So these are the different cycas species we collected from different parts of the Tamil Nadu, as well as when I visited the University of Calcutta, we collected the coralloid roots of Zamia, Dayun, and so on. So this is the Cycas rumphiae. Actually, this cycas we collected from the North Andaman Island in the coastal region. When we are doing the marine cyanobacterial survey, we collected this particular plantlet of Cycas rumphiae, and then we planted in our university. So this is originally collected from the North Andaman Islands. And if you see this coralloid roots, it is a very bunchy one. And if you're taking the cross section, you can see the cyanobacterial zone. So we isolate the cyanobionts, morphological type of isolation, purification. We have nearly 19 different strains. In addition to this, we make the sequencing analysis also for and read the phylogenetic studies. And the next plant we used is the lichens. In lichens also, the cyanobionts are component of the symbiotic with fungus. It is called lichens. There are phycolichens, that is microalgae and fungi. It is phycolichens, and cyanobacteria with fungi is called cyanolichens. So we are interested to work on the cyanolichens. So far in our country, nobody knows what is the cyanobiont, actual cyanobiont residing in the lichen. They said only the cyanobionts, because it is very difficult to isolate the organism from the lichens. So one of my second PhD student, he successfully isolated the cyanobionts from the lichens, and then he maintained the cultures, and he did all the molecular taxonomy and other biotechnology works in this PhD work. And we collected the lichens from Yercaud and Colli Hills. So totally 42 folios, 26 crustose and 9 fructicose lichens we collected from this. Among these, 5 are cyanolichens. So here I gave the list as 5 are cyanolichens. We worked very extensively on the cyanobionts and the cyanolichens in the molecular tools, and these are all the different lichens, and this is the cross-sectional view of the lichens. You can see the zone of cyanobacteria. It is uniformly distributed, unlike the other phycobionts. Cyanobionts are uniformly distributed throughout the cortex of the lichen. And this is 5 isolates we made. And another symbiotic association we worked on is azolla. That is a type of pteridophytes. So in azolla, there is cyanobionts and bacteriobionts are there. Again, people say that only Anabina azolla is present in azolla. But in our studies, we identified more than 100 different strains of cyanobacteria and bacterial symbionts are there using the metagenomic approach. And the first step is the DNA isolation. I'm not going to detail about this. In any kind of DNA isolation, there are 3 major steps. One is break open the cells by using the glass powder and SDS and phenol. So open the cells and get the protoplast. From this protoplast, there is a extraction of DNA using the phenol, chloroform, and chloroform. And final step is the precipitation using the sodium ammonium acetate with 100% ethanol, and later on precipitate it and store it in TE buffer. So this is the major 3 steps in almost all the DNA isolation protocols are there. And after this, we have to go for the electrophoresis, gel electrophoresis to confirm that you isolated the DNA. Genomic DNA. So since the genomic DNA is high molecular weight, it should be very near to the well where we loaded. then we confirm that it is a genomic DNA. If the same bands are coming from away from the well, we can say that it is a broken fragments or sometimes the plasmid DNA. So this slide shows that the different types of gel electrophoresis. One is acrylamide gel electrophoresis. It is useful for the sequencing reactions, and agarose electrophoresis is useful for the localization of the DNA, genomic DNA, and pulse field gel electrophoresis. This is another type of electrophoresis useful for high molecular weight DNA separation. So coming to the PCR, I'm not going to very detailed because everyone knows about the PCR. So it requires the Taq polymerase, it is thermostable polymerase, and then primers. It is only deciding factor for where to start amplification and where to stop the amplification techniques. And Carey Millis was the inventor of PCR. And PCR, it is just making the Xerox copies of DNA, any DNA, supplied as a template, and then starting with the original copy, almost infinite number of copies. For example, if in 0.5 ml tube, you can generate nearly 1 crore of amplified products in this small tube. And amplified fragments can be sequenced, cloned, probed, sized using the electrophoresis. And these fragments can act as the genetic fingerprints. So now we'll go to some other aspects, particularly the details about the PCR amplification. This is the PCR mixture. It requires the template DNA, PCR buffer, forward and reverse primer. These two primers are the deciding factor to start the amplification, and then DNTP mix, Taq polymerase, and so on. And the thermal profile, it is very important for the amplification process. First is the denaturation, so break open the two DNA strands using 92-degree temperature and 60 seconds, and the primer annealing 54-degree or 60 seconds. So the primer annealing depends on the length of the nucleotides present in the primers. If it is a higher number, so the primer annealing temperature will be very high. Whereas in the case of the RAPD fingerprinting, the primer annealing primer length is very, very less than 10. The annealing temperature will be very less. And then polymerization, this is the very crucial step to amplify the DNA. 72 degrees Taq polymerase is very active, and we can generate the n number of fragments. And this is how, for example, this is the genomic DNA. So our forward primer and reverse primer go and bind only exactly with the complementary region of the 16S rRNA, not the other places. That's why we are getting the only 16S rRNA products during the amplification process. And this is how the PCR reaction takes place. So we initially have the double stranded DNA, and in the first step, as I said, the melting temperature, that is 94 degree, the two strands will get separated. And next is the primer annealing. The temperature is very less, 50 degree, where the primers go and bind with the complementary region of the opposite strands. Here you can see this. And third cycle is extension, that is 72 degree temperature, where the Taq polymerase binds in this double stranded region, and it add up the nucleotides present in the PCR reaction tube, and it gets the extension. Likewise, in the first step, you are getting the two fragments of DNA. And the second step, again, it is repeating. 92 degree, gets two strands gets separated, and then again primer annealing. The same process is continuing. And then in third cycle, this is very important cycle. You can see this two fragments are get separated by 92 degrees or 94 degrees, and annealing is 50 degree. Again, it goes match with the complementary regions. And you can see this after amplification. So this is the fragments of the defined size. So only 1600 means it is 1600. During the course of the amplification, thermal variations, 92 degree, 54 degree, and then 72 degree, the extended region will automatically degraded. So likewise, finally, we are getting only the double standard 16S molecule of exact size. So if you see this number, first cycle it is only 2, only single strand, and second cycle 2 fragments are there. In third cycle 4, and eighth cycle, then fourth cycle 16, and then fifth cycle 32, and then sixth cycle 64. Likewise, if you are going for the 25th cycle, there are around 83 lakhs of fragments of amplified products you are getting in the F&F tube. So this is the need for the further analysis, as well as the cloning, sequencing, etc. And once you amplified the DNA, amplified the particular 16S rRNA, you have to identify, we have to confirm whether we are really amplify the DNA 16S rRNA product or not. For this, we have to run a gel with a marker, and you have to add the positive control and negative control in addition to our selected DNA from the symbiotic cyanobacteria. So positive control is needed, because if any problem with the PCR kit, you cannot get any bands in this. So positive control is confirming the activity of the PCR kit. Everything, tag polymerase, other components are very perfect. The negative control is equally important, because if you have any negative control, we are not adding any template DNA. Instead of, we are adding all the components except the template DNA, and this negative control, there should not be any band, because there is no template DNA. If you have any contaminated DNA, any contamination, if you get any bands, that means your DNA, your PCR kit is not in a pure, or sometimes some contamination will be there. So we cannot see that, say that it is a 16S rRNA of our own selected DNA. So negative control, there should not be any band. And then these are all our actual DNA, and ours is nearly 1,600 base pairs. We just match the DNA ladder, and we confirm that it is a 16S rRNA product. And the next step is the ligation. As I said, if you want partial sequencing, after confirming the gel, you just send the sample, amplified product, to the sequencing company, and then get it sequenced. But you want all the 1,600 nucleotides. So you go for the cloning, that is the ligation reaction, in which you have to ligate your amplified product with DNA vectors commercially available. And again, once you ligated, you have to confirm using the gel. So for here the marker, and this one is only the vector DNA. And if you clone with the vector DNA with the 16S rRNA, definitely the molecular weight of the cloned product is very higher. So this is the, from this second lane, we confirm that it is the vector with the plasmid DNA. It is a recombinant DNA molecule with the PCR product. We confirm it, and the next step, go for the transformation. So this is the just details of TA cloning. So this is the vector we used. That is PCR21. There are so many vectors are commercially available. It is a kind of linear vector initially, and it has the T overhangs. This vector has the T overhangs within the lag Z site. Okay, so our PCR product is this, and one specialty of the tag polymerase we are using, it has the template independent addition of one adenine in the 3' end. So based on this, the vector DNA has T overhangs, and our PCR product has A overhangs. So TA cloning is possible because it is exactly matching with the complementary nucleotides. So this PCR product will be inserted within the lag Z site. Okay, so the lag Z, the galactosidase enzyme site is disturbed when we are inserting the PCR product. So this factor is useful for the markers for recirculation of the plasmid or recombinant plasmid with the PCR insert. So here, after this, you have to transform the cloned product with the E. coli JM109 competent cells. It is ampicillin sensitive, and the other genetic markers are well defined. So we select this JM109 competent cells and mix with the ligation reaction, the cloned product. And then after this, from this product, we add the cloned product with the SOC medium with 37 degree with gentle shaking. After this, we played this 100 microliter of the SOC medium containing the plasmids, recombinant plasmid. We played this on the LB plates containing ampicillin, Xgol, and IPTG. Because this is actually useful for segregating the recirculated plasmid and recombinant plasmids, and incubated overnight, and then we are getting the blue-white colonies. So this is how we are getting the blue-white colonies. Here, the blue colonies represent the recirculated plasmid without our PCR insert. The vector, the PGMT vector or the other vector we used, it is recirculated. Because of the recirculation, the beta galactosidase enzyme site is very active. So here we add the in the plates we add the Xgol and IPTG. Because of the undisturbed galactosidase enzyme site, they utilize the Xgol and IPTG. It forms the blue colored colony. Whereas in the white colored colony surrounding with this plate, because the vector is disturbed or inserted with the PCR product, so the beta galactosidase site is affected. So it cannot utilize the Xgol and IPTG present in the medium, agar medium. So it won't produce the blue colored colonies. Instead, we can get the white colored colonies. All the white colored colonies, we says that it is a plasmid with PCR insert. So we just make the library like using the clones and the number of numbers we included, and then we just transfer each and every colony in these plates, and this is the library of the cloned product. So each and every one represent this one. So initially in our amplification, particularly the Azolla genomic DNA, we use the crude Azolla genomic DNA. So here we are using the universal prokaryotic primers. So it amplify only the Azolla, only the prokaryote, that is bacterial and cyanobacterial DNAs. Whereas the eukaryotic Azolla DNA will not be amplified because it is only specific to the prokaryotes. So once you have the prokaryotes, prokaryotic amplified product, here the source of 16S rRNA is different. Some amplified product from the cyanobionts and some amplified products from the bacterial symbiotic bionts. So we have to differentiate which one of the white colony having the cyanobacterial 16S rRNA and which one is the bacterial 16S rRNA. For this, we have some blotting techniques also. So once we develop this a library of different clones, go for the plasmid isolation. I'm not going detail about this plasmid isolation. So everything is standardized, the protocols are available. Once you have the plasmid, now you have to differentiate which clone is having a cyanobacterial origin, which one is a bacterial origin, for which we are using the cyanoprobes, cyanobacterial specific probes, that small nucleotides sequence which is tagged with fluorescent pigment, fluorescent chemical, and then you use this, isolate the plasmids and use this dot blotting. So here we use only the zeta probe membrane, and in which we loaded the plasmids of different clones. So here, after transforming the plasmids and go for drying, and some special procedures are there. Once drying, you use the cyanobacterial specific primers in the solution, and then you make some small agitation with the tub. And wherever the cyanobacterial 16S RNA is there, our cyanoprobe go and binds. And it because of the dye, you can get the blue colored dot. So blue colored dots are nothing but the cyanobacterial 16S RNA, whereas the empty, there is a white colored or no blue colored dot is there. So that represents the bacterial origin, 16S RNA of bacterial origin. Likewise, we can differentiate which one is the cyanobacteria and which one is the bacterial clones, and later on we select the different clones and send it for sequencing or do the manual sequencing, and we can make the phylogenetic tree and identification. Everything is possible. So coming to the DNA sequencing, again, this is for example, this is the recombinant plasmid with the PCR insert. So now we are going for the thermal sequencing technique. Just like a PCR, we add all the primers, that is sequencing primers. The sequencing primers starting from the thermal region of the vector site, from the 5' end and as well as the 3' end. This is our PCR product. And this is the cloned, this is the our actual gene of interest. At the end of the vector, it has the primer binding sites. We add the sequencing primer in the opposite region, and we are using the thermal sequencing. Here, it is a special type of sequencing mixture. In addition to in PCR, we are adding the deoxynucleotides, whereas in sequencing reaction, in addition to the deoxynucleotide, we are adding the dideoxynucleotides for chain termination process. So I will give it in detail in subsequent slides. So once it is bind, and because of the PCR reaction, again it is amplified. And this is how the difference between the deoxynucleotides and dideoxynucleotides. Here in ribonucleotides, there are two OH molecules are there. In dioxynucleotide, there is single OH molecule, and dideoxynucleotides, there is no OH molecule is there. So because of this absence of OH, the amplification, that is the extension is terminated. That is called chain termination reaction. In the reaction mixture, if one complementary region of the DNA, it utilizes the deoxy, that is DNTP, the chain elongation takes place. Whereas instead of the DNTP, the dideoxynucleotide is selected and bind with the complement region, there is no possibility of extension. The chain is terminated. So likewise, we are getting the different molecular weight fragments in the DNA. And this is how the sequencing reaction. For sequence reaction, we have to, this is what I am telling about the manual sequencing. When I was in US for 1 year, I did manual sequencing technique for sequencing of cyanobionts as well as the bacteriobionts. So we have to use 4 different tubes. All tubes having the template DNA, all the PCR reaction mixture. In addition to this, it has dideoxynucleotides and deoxynucleotide in one tube. Likewise, all the 4 tubes, all dideoxynucleotides of respective nucleotides will be added. 4 tubes. Okay. So here the 4 reaction setup, each containing, as I said, DNA polymerase enzyme, primers, template to be sequenced, DNTPs. In addition to this, a small amount of 1-dideoxynucleotide, that is DNTP, that is ATP, CTP, GTP, and TTP. All are dideoxynucleotides should be added, and incorporation of dideoxynucleotide terminates the DNA replication. So we are getting the different molecular weight fragments that reflects the sequencing reaction. For example, if you see this reaction, for example, this is the template DNA, and if you add the primer, this is the sequencing primer, you consider this, and then you add the tag DNA polymerase. If it is a T, is there. If deoxynucleotide of A joins, the chain is extended. Whereas instead of deoxynucleotide, dideoxynucleotide, that is dideoxy A, joins in the complement region, the chain is terminated. So we are getting a small fragment of DNA, terminated DNA. And in addition, in the same primer binds, if in other region, if it is dideoxynucleotide binds with this complement region, again we are getting the terminated fragments. Here 2 different molecular weight fragments are there. In case of the A tube, that is dideoxy and deoxyadenine mixed tube. Likewise, wherever the complementary dideoxynucleotide binds with the complementary T, it is chain is terminated. Likewise, wherever the dideoxy joins, you are getting the, here you see the different molecular weight fragments. This is in the case of deoxy A nucleotide tube. Whereas similarly in G tube, the dideoxy G, dideoxy C, dideoxy T, you are getting the different molecular, each and every nucleotide is terminated. So these features will be helpful for getting the different molecular weight fragments that we sequenced, and we are reading the sequences very easily. And this is how you can see this. Here in the sequencing reaction, there are 4 tubes. That is, I said A, T, G, C like this. So you just load the dideoxy components in this first lane, second lane, third lane. Likewise, you are getting the resolved DNA bonds. So we can read from the base to top, and you can read the sequences. For example, here we are mentioning here. This is the actual sequencing we are mentioning. So this way we can easily read the sequences. This is using the autoradiogram. Here we are using the 35S radioactive molecule for sequencing. Now the automated sequences are there. It need not use the radioactivity. We can use the chromogram for chromophores for the sequencing, tagging all the each and every dideoxynucleotides. We tag with the different colors, fluorochrome, and we can get it the sequencing. And this is the automated sequencing. In automated sequencing, it is somewhat different from this manual sequencing. Here, all the there is no 4 different tubes. All the PCR mixture, the template DNA in a single tube. It add all the DNTPs and dideoxynucleotides based on the molecular weight. After the thermal cycling, based on the molecular weight, it moves from the gel. And in the basal region of the cell, there is a laser detector, and each nucleotide is tagged with a different colored fluorochrome. So if, for example, if T is leading to the fluorometer region, the detector region, so it just capture the color, and we can get the single red colored peak. That means it is a T. And next G, it moves to the base, and again we are getting this colored peak. Likewise, so whatever the molecular weight, based on the molecular weight, it reaches to the detector, it detects the color, and then we are getting the different peaks. Likewise, we can make the sequencing. This is the case of the automated sequencer. That is a next generation sequencing, Illumina sequencing. There are so many techniques and instruments are available for doing this kind of sequencing reaction. So once you have sequenced, you have to match your sequence. This is the partial sequencing of mine, and I matched with, I use the RDP database. I submit my sequences, and immediately we are getting this page. So our sequences is matching exactly with the nostoc muscorum in particular region of the DNA. This is the 1600 nucleotide base pairs, and we can see this. Our sequencing is exactly matching somewhere here. So we can confirm that this is our unknown sequences are having nostoc muscorum sequencing. So likewise, this is the automated sequencing results. You can see this different colored sequencing, and we can match this sequencing with the phylogenetic tree. And once we are getting the sequences, we have to submit our DNA in the gen bank. There are 3 gen banks are there. One is NCBI, EMBL, and DNA database of Japan. All are similar. So if you submit our sequences in the NCBI, the same sequences with the different accession number in EMBL and DDBJ, that is DNA database bank of Japan, we can get our sequences in all the other 2 gene bank. Okay. So if you want to, once you submit the gene bank, there is some procedures are there. There are 2 options, major options in the gene bank submission. One is Bankit, and another one is sequence. If you have 1 or 2 few sequences to be submitted, you directly submit in online facility, Bankit form. You can just go to this website, and then you cut and paste your sequences in the Bankit. Immediately you are getting the acceptance, and then it will process. It will take only within a week we are getting the access number. So if you have a large number of sequences, you just download the sequence format, and then leisurely, whenever time permits, you just load it, and once you completely load it in the sequence format, you can submit it. So only difference is this is the instant submission. It is if you have a very large number of sequencing, you may use the sequence software. So if you go to the Bankit software, it asks only 4 questions. One is the general submission information. What is this? Whether it is a 16S or 18S of prokaryotes or eukaryotes, anything you can use. And then reference information. So what is your name and what are the things should be coming in this one? It is up to your choice. And then source of information. So whether it is a eukaryote, prokaryotes, or what kind of gene you are loading, the 16S RNA or any the pigment genes or any other specific genes you have, and then finally you cut and paste your DNA sequence in the box provided. Okay. Once you submitted this one, in the case of NCBI, you are getting this kind of page, access numbers. You can see here. This is the calothrix. As I said in the cycles, only in Nostoc cyclidae, we isolated and identified calothrix species, a new genera in this symbiotic strain. And this is another one, EMBL. We submitted the sequences of Nostoc. And another one, DNA database of Japan. Likewise, we are getting your sequences in the GenBank also. Once it is over, you have to make the phylogenetic analysis. There are so many phylogenetic methods and softwares are available. I'm not going to detail about this. And these are all these free softwares available for making the phylogenetic analysis of POP, Mega, Phylib, and so on. So once you use this software, particularly Mega software or Phylib, you can see this phylogenetic relationship. For example, this is from the cyanobionts of the lichens. Cyanobionts of the lichens, because it is totally new to the science. Nobody so far sequenced. Our cyanobiont sequences are having a separate branch. It is not inserted with any other branch, because it is a new kind of sequences we deposited. This is the first time in our country we isolated the cyanobionts from the lichens. That's why it is in a separate branch. And another one, it is the cycles. As we said, is different type of cycles isolates of cyanobacterial symbionts. So this is just initiated with two of our sequences like Nostoc species, calothrix and Nostoc, and this is how we are our isolates are closely related with the Nostoc, and other calothrix is related with the calothrix brevissima. Likewise, we make the phylogenetic analysis. And this is the the metagenomic approach of cycles. Since I used the prokaryotic primer, so our clones are matching with the heterosisters branch, the 513. These are different clones we sequenced. So it is all grouped together with the heterosisters branch. So in addition, in the cycles, there are so many bacterial bands, more than 500 bacterial clones we got it. And the bacterial clones, we initiated with 5 bacterial clones. All the 5 bacterial clones are distantly related, not the similar one. Likewise, we can differentiate and distinguish the biodiversity using the primers metagenomic approach. And another type of fingerprinting is RAPD fingerprinting method. Because time is already exhausted, I'm going very shortly. The RAPD and the 16S is based on the length of the primers. In RAPD, less than 7 or less than 8 sequences, nucleotide sequences in the primer. Because of the very small number of nucleotides, it randomly join with the complementary region of the DNA. So there are so many number of fragments we will generate. On the base of the DNA bands, getting the very small primers, that is randomly amplified polymorphic DNA primers, we are getting the different types of bands. So this is helpful only for differentiating the closely related species, not for the identification, as I said. And RAPD fingerprinting, if you see this is, for example, this is template DNA, and because of the very small length of the primer we used, it randomly binds with the complement regions. And almost all the binds region is not capable of amplification. It requires some rules. For example, here the template is binds many locations. And you see this one, the primers away from each other, so amplification won't access. Likewise, there are some rules are there. Here also, the primer points in the same direction, so there will not be any amplification. And then similarly, and here you can see this, the primer amplification is just 100 to 1500 base pairs. The amplification will be there. So based on this rule, you are getting the different number of bands. This is, for example, we isolated nearly 19 strains of cyanobacteria in the Cycas coralloid roots, and we made the RAPD fingerprinting techniques. You can see this, even though all are nostoc species, but the genetic band makeup or bands are different from each other. So this is the molecular weight of the previous slide gel. Even though here you can see the bands, number of bands are similar in different clones, so different strains of 14, 14, 14 like that, and the molecular weight of the each band is different. So likewise, we can proclaim that even though it is the same nostoc species, but the genetic makeup will be vary. So based on this variation, we make the phylogenetic tree also using the RAPD banding pattern. So this is also possible. And another very interesting study we made is we are using the same species of different isolates. For example, we selected six Synechococcus elongated species, but it is isolated from the different geological locations. That's why the strain number is vary. Even though the morphologically they all are same, that is Synechococcus elongatus, but if you are making the RAPD fingerprinting, the banding pattern will be unique. This is the rep primer we used, and this is the STR or SAT randomly repetitive sequencing amplification. You can see the distinct banding pattern also. So from this, even though we conclude that even though the genus and species is same, but the geological location where it grows, where it survives, the genetic makeup will be different. So that's why if you identify the particular region, we may use it as a barcoding of the sequencing. And here, if you have the 16S or any amplified product sequencing, if you use the gene B structure prediction software, just cut and paste your sequence in the software, in no time you are getting the secondary structure. By seeing the secondary structure, we can distinguish this one. Each of the secondary structure is different. Nostoc species is different. You know the different because of the complementary region. You can get the stem, and uncomplementary region you are getting the loop-like structure. Likewise, each and every product DNA of 16S RNA is differ. By this way, we can differentiate the closely related species also. And in addition, there is another New England Biolab, that is NetCutter software is available. This is the RFLP method. We need not use the restriction enzymes for the cutting the DNA in the specific region. Instead, this NetCutter software, you just download the software and then you cut and paste our genomic DNA. This is our genomic DNA, and the software will tell wherever the restriction enzyme sites, the restriction enzyme site of nostoc species, and this is another restriction enzyme site of the nostoc species. You can see the variation in the restriction sites. So based on this, we can differentiate. It is freely available software. Anyone can use this if you have the sequence. And so academic point of view, we made the protein fingerprinting also. Protein fingerprinting, all the isolated Cycas cyanobionts, you are getting the different distinct banding pattern, but it is not constant. If you change the pH or change the any environmental condition, definitely you are not getting the similar kind of banding pattern. And by academic curiosity, we did it, and based on the protein fingerprinting, so we can make the phylogenetic analysis also. But it is not perfect because you can see first one is haplosyphon. It is a heterocystus branched form, heterotrichous form. It is a filamentous form, non-heterocystus, but both are in the same clade. So this is not useful for the identification. And another extensive study we made the another type of RAPD, that is M13 based genotyping of marine cyanobacteria. We published this paper in our Journal of Applied Phycology. You can see the genetic makeup of each and every isolates we are maintaining in our marine cyanobacterial germplasm. And coming to the barcoding, so barcoding is now a need of the hour to identify exactly the strain level. So this is very important. DNA barcoding is a short DNA sequences from a uniform locality. It is very important. Uniform locality on the genome used for identifying the species. And Paul Hilbert in the University of Gulf, Canada, he proposed DNA barcoding of animals using the mitochondrial cytochrome C oxidase, that is COI genes. And this is very efficient. And for plants, the mitochondrial genes do not differ sufficiently to distinguish among the closely related species. And instead, they use some chloroplast genes for the DNA barcoding. And why barcode of animals with mitochondrial DNA? Because following attributes. So one is greater differences among the species. For example, in genomic DNA, the variation is 0.9, and whereas in the mitochondrial DNA of the same species, it is 9% differences there. So differences sequences are needed for looking the barcodes. And then copy number, there is a high copy number present in the mitochondrial DNA. Instead of nuclear DNA is only one or a few per cell, and relatively few differences within the species. And the introns, which are non-coding sequences in the genomic DNA, whereas it's absent in mitochondrial DNA. This is another attribute why we are using the mitochondrial DNA in the case of animals, eukaryotic animals, for DNA barcoding. And there are several barcoding softwares are there, particularly Bold System of Canada, that is Barcode of Life system in Canada. It is freely available. So if you have any sequences of specific region, if you send the sequence to the Bold system, it will make the barcode like this. So we make the barcode also, and you can see this Barcode of Life systems. This is the website that is freely available. So initially we isolated the fungus and sequenced the ITS sequence of the fungus. We submitted this ITS sequences to Bold system, and we are getting the barcode of this one. Now we have standardized the barcoding technique for eukaryotes. Now we are applying this technique to cyanobacterial germplasm available with us. And we made several barcodes using some fungi, indigenous fungi isolated from our university, trichoderma, different penicillium species, and we easily barcode this fungus using the barcode of life data system. This is the barcode we can use. And as a result of our total molecular taxonomic studies, we made 16 RNA of cyanobionts and bacteriobionts or cyanobacteria, and 18 rRNA of microalgae and fungi, and then group 1 cell splicing introns, and so on. So far, nearly 594 sequences we deposited in the gene banks from marine cyanobacteria, freshwater, symbiotic cyanobacteria, actinomycetes, bacteria, subsurface bacteria. This is actually new to our country. This is the first report on the subsurface bacterial sequences, then fungi, microalgae, microzooplankton, viruses, and group 1 introns we submitted so far. Now we have the very good, excellent germplasm facility in the name of National Repository for Microalgae and Cyanobacteria. Now we are going to make all the nucleotides as a barcode of these isolates. So this will be of useful in future. And coming to the conclusion, based on our experience, morphological characterizations is very, very important. It is base for any taxonomic aspects. So biochemical features are needed in the case of some of the bacteria. And as I said, it is not useful for the identification because of the change of condition, change of pH. Definitely the biochemical features will vary. And the molecular characteristics is very important, particularly the 16 rRNA sequencing is meant for useful for the identification, making the file identically, and so on. The other fingerprinting methods like RAPD, RFLP, REP-PCR, LPR, PCR, these are all useful for distinguishing the close related species of close-related species, not for the identification purpose. So this is really the important tools to study the genetic level variations, distinguishing between the isolates of the same species, provide more reproducible results than the other methods. So this is what, even the morphological characterization, if you're collecting the cyanobacteria or any microalgae in the field, the morphology is entirely different. If you are repeatedly subculturing in the culture medium, because of the availability of nutrients throughout the year, it loses its wild characteristics. For example, in nostoc, in nature, it is like a mucilaginous ball. So because of the stress condition, in the absence of water, the nostoc producing the mucilage around this, and then it imbibe the water and keep the water for the longer period. So in morphologically, the nostoc means it is a mucilaginous ball in which the filaments are embedded. Whereas the same mucilaginous ball like nostoc, if you are growing in the laboratory condition and the repeated subculturing, because of the availability of the nutrients throughout the year, this nostoc won't produces the mucilage. Instead of it looks like anabina. So it gives a misleading. For this, the molecular tools, particularly 16S rRNA, is very, very important to identify the genus and the species, even in the strain level variation. And with this, I thank my Guruji, Dr. G. Subramanian, who is really insisted me to work on the taxonomy of microalgae and cyanobacteria. Initially, I hesitated, then later on, I knew the importance of the molecular taxonomy and the conventional taxonomy. I did it. And now I'm receiving the cultures from other countries also for identification of cyanobacteria and microalgae. I also thank Dr. Sandra Nairviki Bauer, where I learned all the molecular taxonomic techniques through the DBT Overseas Associateship Award for long time award. I stayed in the laboratory and learned all the molecular based techniques. And thank Dr. Ukureti, Lichenology Department, for identification of our lichens. Dr. Abubakar, helpful for identification of the cycles, and Ramamurthy, he is the person who is initially sequenced all our 16S rRNA and 18S rRNA gene sequences in his laboratory. And I thank the Department of Biotechnology and Department of Ocean Development. Now it is Ministry of Earth Science. So with this, any doubts or clarifications, now you feel free to ask.
Thank you very much, sir, for your wonderful lecture. And there are some questions posted by the participants, sir. Shall I?
Yeah, you can ask the questions.
Yes,
just read out the questions.
Yes, sir. So one of the undergraduate students have asked, is there any book or manual is available for identification of the algae?
Actually, in our National Repository for Marine Microalgae and Cyanobacteria in the university website, so we made some key factors, and taxonomy of freshwater and the marine cyanobacteria is available. And there are a large number of monographs available in terms of algae, particularly TV Desiachari, Cyanophyta, and so many other foreign scientists also contributing a lot for identification of microalgae and cyanobacteria.
Yes, sir. Thank you, sir. And one more question posted by the PG student. What contaminants loss of resolution in plasmid or the DNA Sanger DNA sequencing method?
What is it?
What contaminations cause loss of resolution in the DNA sequencing methodology?
Actually, in DNA sequencing, I showed the autoradiogram. That is autoradiogram. But initially, some of the sequences are very faint, and it goes down because of very low molecular weight. And in between sequences, it is exactly looking very visible. And topper sequences, which it is very contested. Okay. For this, we are loading the sequencing, same sequencing reaction in 3 different. So for example, you load 10 o'clock, and then it sequenced for 3 hours. Total 3 hours if you use this. 10 o'clock we are loading sequences, and 11 o'clock and 11:30. Likewise, we different intervals we are loading the sequences. So based on the mobility, in 3 hours, the thicker region initially in the top, and in between, the other sequences will come and clarify. and very small sequences you can get it in the very clarity in the base. So likewise in 3 different duration, the same sample if you load, you can get the very clarified one. It is not a contamination, it is because of the molecular weight as well as the mobility. As you see in my autoradiograph, in the topper region is very congested. It is very difficult to read. For this, we prolong the duration. So smaller nucleotides go to the bottom and go to the gel, and the topper region comes down. So we can very clarify easily. Likewise, 3 or 4 durations are needed to read the entire sequencing.
Yes.
I think I answered his question.
Yes, sir. Yes, sir. So another question posted, like how to choose the primer for RAPD analysis.
So it is commercially available. So you can any reference it is there, or otherwise you also design based on your requirement, because it is not specific for any gene. It is not specific for any gene. It is a small oligonucleotide primers, 5 to 6. Even you can just imaginarily you just make the sequence of your own interest and develop this small, because very smallness, the probability of the presence of particular nucleotide is very high. So it is commercially available also, in literature it is also there, and you can design your own also, because it is not represent any gene.
Yes, sir. And myself have one question, sir.
Mm.
Sir, does this barcoding method can be employed for conservation of the endangered species or identification of exotic species, sir?
Yes, yes, definitely. That's why barcoding is very, very essential. But if it is an endangered species, we have to isolate the DNA and identify the marker DNA to barcode. So once you have, definitely if you send the sequences, immediately you are getting the barcode.
Okay, sir.
It is possible.
Okay, sir. So I think there are no more questions, sir. So I wholeheartedly thank you very much, sir, for your fantabulous deliberations. I'm sure the research work and the activities that you have discussed with us will be a great inspiration to all the students and the scholars to pursue their higher studies in the field of cyanobacteria and other related algal studies. So once again, I thank you very much, sir, for your presentation.
I also thank you for your nice conduction of this seminar or workshop, and also I thank Dr. Tapan Kumar sir and Dr. DJ Bakira sir for suggesting my name and give a platform to share my expertise on taxonomy of microbes. Thank you, and all.
Yes, sir. Thank you, sir. So dear participants, we are breaking here for tea. So we'll be joining at 11:30 a.m. sharply for the technical session 6. So kindly join us at 11:30 a.m. in the same YouTube link in which you are here now. So at 11:30 a.m. we'll meet you again. Thank you all for the support.
ಹೋಗ್ಲೇಬೇಕಾ?
So, welcome back the participants. Now we are in the technical session 6 of day 2 of Science Academy's lecture workshop on recent trends in microbiology and biotechnology. So now I request Dr. D. Siva, Assistant Professor, Department of Biotechnology, Srimad Anand Arts and Science College, Trichy, to introduce the speaker of the session, Dr. D.J. Bhagiraj, an ASI Honorary Scientist and Chairman.
Good morning to one and all gathered here. I'm very happy to welcome you all for this 2 days Science Academy's virtual lecture workshop on recent trends in microbiology and biotechnology. And it gives immense pleasure to introduce Dr. D.J. Bhagiraj sir, NASI Honorable Scientist and Chairman, Center for Natural Biological Resources and Community Development, Bangalore. He has completed his postdoctorate at New Zealand, Australia, UK, and USA. He received various fellowships such as S.R. Vyas Memorial Award, Dr. G. Rangaswamy Memorial Award, Dr. T.S. Sadasivam Memorial Award, Dr. V. Agnihotrudu Memorial Award, Dr. R.Y. Rai Memorial Award, Dr. Nadarajan Memorial Award, and he is a recipient of Distinguished Asian Mycologist Award 2019. And to his credit, he has completed 400 research papers, and he has published 101 review articles, 11 books, and he has completed 36 research projects as principal investigator in microbial inoculants for sustainable agriculture. And he is being an advisory committee in ICAR, DST, DBT, CSAR, UGC, ICFRE, NBA Rubber Board, IFS Sweden, AICR Australia, FAO, and UNEP. And in specially, he has honored for his contribution. A new AM fungus has been named as Glomus Bhagiraji, and he has completed 23 PhD students. With this brief introduction, I hand out the session to Dr. D.J. Bhagiraj sir, and thank you, sir. Welcome you, sir.
Yeah, thank you very much, Dr. Siva. So it's nice to again address the students of your college. I think this is the 3rd time or 4th time.
Yes, sir.
To their college, a beautiful college campus. And so today I'll be talking on arbuscular mycorrhizal fungi. Host in there. Huh?
Please make the sir as the host.
Yeah, sure. Sir, host. Sir, host. Yes, sir. We made.
Okay. Okay, we can go to the slides now. Slide. Okay. So today I am going to talk on arbuscular mycorrhizal fungi for sustainable agriculture, horticulture, and forestry. So as microbiology students, I am sure that you're all aware of the word mycorrhiza. The word mycorrhiza stands for myco standing for fungus, rhiza standing for root. So it shows that it is an association between plant roots and certain fungi. And it was coined by a scientist by name Frank, and in the year 1884. And it has a very interesting history behind it. And Dr. Frank was a person who was interested in forest pathology. That means looking into the fungi associated, pathogenic fungi associated with forestry species. And once when he went and he saw that the roots of certain pine seedlings were covered with fungal mantle. So he thought it must be a pathogen. He brought it to the lab, isolated the fungus, and he wanted to prove the cause postulates. That means to prove the pathogenicity. So he inoculated healthy pine seedlings with the fungus what he has isolated from the diseased or the roots covered with the fungus. So he was waiting that the seedlings will become diseased, but the opposite happened. Those inoculated seedlings started growing better. So then he thought it must be a beneficial fungus and not a harmful or a pathogen. So that is how he coined the word mycorrhiza. And further, he also said one more statement that it is a symbiotic association between plants and certain fungi. That was in the year 1884. And after that, not much work has been done till almost 1950s. And here in 1950, a student from University of London was asked to select a thesis problem for her PhD work. And so she was looking at what she can work on, something new. And her name was Barbara Mosse. And she saw that mycorrhiza was described by the German scientist, but not much work has been done. So she decided to work on mycorrhizal association in apples, which is a very important fruit tree in England. And her thesis she submitted, and then later joined the Rothamsted Experimental Station in England, which is considered to be a mecca of agricultural scientists. And she continued her work on mycorrhiza. And in addition to her work, she invited people from any part of the world interested in mycorrhiza research to her lab with the fellowship. I would like to underline that, with the fellowship, so that they can come and learn the mycorrhizal association in croplands and go back to their country. Thereby the research spread to, that is how the research can spread to different parts of the world. So that good thing she did. And that is the reason why we call Dr. Barbara Mosse as mother of mycorrhiza research. Many branches of science have fathers. Very few branches have mothers. So mycorrhiza research has a mother. And when we had the international conference on mycorrhiza in Bangalore, I invited her to be the chief guest to give the keynote address. She was good enough to come and give the keynote address here. And now we have four main types of mycorrhizal association. Ecto, vesicular arbuscular, now called as arbuscular mycorrhizal fungi, ericoid mycorrhizal fungi, and orchid mycorrhizal fungi. And ectomycorrhizal fungi are commonly associated with temperate forest tree species. Ericoid mycorrhizal fungi are commonly associated with the plants belonging to the family Ericaceae, which is again commonly found in temperate regions. We find few ericaceous plants like Azalea, rhododendron in the Himalayan region of our country. And the fourth one is orchid mycorrhiza. As the name itself suggests, that's very, very specific to orchids. And the fourth category, which is important in tropical countries and commonly associated with crop plants, is the vesicular arbuscular mycorrhizal fungi. Earlier, they were called as vesicular arbuscular mycorrhizal fungi. Now the name has been changed to only arbuscular. I will tell you a little later why it has been changed. And here you can see this is the transverse section of a non-mycorrhizal root. You can see the epidermis and the endodermis and the cortex region in between. And this is a root, transverse section of a root colonized by the arbuscular mycorrhizal fungi. The fungus lives in soil and produces very large spores called as extramatricular chlamydospores. The fungus penetrates the epidermis and then spreads in the cortical region. And in the cortical region, it produces globose to oval spores structures called as vesicles, and then highly branched hyphal structures called as arbuscles. So it produces vesicles and arbuscles. That is why they were named as vesicular arbuscular mycorrhizal fungi. Later, it was found that few species, or not even species, few genera of ampfungi do not produce vesicles, but all of them produce arbuscles. That is the reason why the word vesicular was deleted. Now they are called as arbuscular mycorrhizal fungi. So if you see the old literature prior to 1990s, you will see that they were called as VAM, vesicular arbuscular mycorrhizal fungi. Now later, recent references, you will see them as arbuscular mycorrhizal fungi. And most of the tropical plants or crop plants important in agriculture, horticulture, forestry, are all colonized by this fungus. So I'm sure that if you just go outside in your college campus, try to bring a root sample of the plant growing there, and then follow the staining procedures, you will see the vesicles inside the root system. It's a very common thing in all the tropical countries. And these are the arbuscules. Arbuscules are highly branched hyphal structures. They are dichotomously branched, but they are short-lived, 4 to 5 days. They undergo degeneration. That's why we don't see this branching very clearly many a times when we do the staining. So vesicles and arbuscules are the characteristic feature of most of the genera, but all of them produce arbuscules. All these fungi belong to the phylum Glomeromycota, which has 3 classes, 5 orders, 14 families, and 26 genera. And as it stands today, it's about 230 described species, as the taxonomists are adding few more species now. I said there are 26 genera of ham fungi. Of these 26 genera, which are the most commonly occurring genera? And therefore, the most commonly occurring genera are Glomus. A is Glomus, B is Gygospora, C is Oculospora, and D is Entrophospora. So these are the 4 genera of ham fungi occurring very frequently throughout the world, including India. The next question, if we ask, which is of the 4, which is the most frequent genus? And the answer is A, that is Glomus. So Glomus is mostly present in almost all tropical soils. So again, I can say if you just take the soil sample from your campus and do the wet sieving and see the scores under the microscope, I am 100% sure that you will get Glomus. So it is so commonly present in our soil. And these fungi are obligate symbionts. That means we cannot grow them in culture medium, just like PDA or Zepex agar or Richards medium, like any other fungi. So they need the roots of a living plant to grow and multiply. So that is why we maintain them as pot cultures. So here, this is how we maintain them in our lab. So here it is maintained in the glass house, and you can see each row is one particular species of fungus, ham fungus. And this again takes back to the history. This is a slide which was made in 1977. So I had a Fulbright fellowship for 1 year to go to US and work on microbial genetics. So the object with which I was to work was to transfer the nitrogen fixing genes, the NIF gene, from Rhizobium to a saprophytic bacterium, that is Erwinia herbicola. So I landed in US, University of California, Davis campus, and then it was with the Professor Mortimer Starr, who was the editor-in-chief of Annual Reviews of Microbiology, very great person. So I started the work. The next lab was working on mycorrhizal fungi. I used to peep into the next lab. I somehow felt the mycorrhizal research was more interesting than my transferring NIF gene. So I went to Professor Starr and asked him, can I shift to the next lab? Then he said, you have come on a Fulbright fellowship. The Fulbright fellowship office is in Washington, DC. You talk to Washington, DC people, and if they don't have any objection, and if the other professor, the mycorrhiza professor, has no objection, I have no objection. So I called the lady in charge in Fulbright office in Washington, and she said, no problem if both the professors agree for your change. Then there was no problem. I shifted to the next lab working on mycorrhiza. There is a very common saying, that next house curry or sambar tastes much better than your own homemade sambar. So that happened also to me. So when I shifted to mycorrhizal lab, there was an intensive work. I was trained in mycorrhizal research, and it was for 1 year. The Fulbright fellowship was for 1 year. So it was a December 31st. My fellowship was coming to an end. And in October, the professor asked me, do you want to stay in US for another 5 years? We can take you on a contract basis. I was a bit worried what answer I should give him. I was married at that time, so I called home. No mobile those days. You have to call. And then I asked my mother and my wife. Both of them said, come back. And I also talked to the Indian scientists who were in the campus. And all of them said, we are all sticking on here in US because we never had a permanent job in India. You are already an associate professor in a government university. You should go back. So I decided to come back. Many people said I am a fool taking a decision like that, but I don't think I did a wrong thing. So when I came back to Bangalore, we never had any information whether the Bangalore soils have mycorrhizal fungi, whether the plants are colonized by mycorrhizal fungi. I told my students, just go and bring some plants, let us stain and see. And this is what they stained and showed me. You can see 3 different crop plants, all colonized by mycorrhizal fungi. The interesting thing here is, many of the reports, what we made, became the world first report, because the temperate countries do not grow what the crops we grow here. You know, many crops are here which we grow, and like clove, cardamom, ragi, all became the world first report. And when I came back from US, the professor asked me, his name is John Mengi, he asked me, what do you want to take back to India? I told him, the best mycorrhizal culture you have. And the best mycorrhizal culture they had was Glomus fasciculatum, which they used to give to the farming community for growing apples, avocados, citrus, and so on. So he gave me that culture, which I brought. And this was the first experiment we did. This was done in Bengal gram. You can see the difference. And we used in this study sterilized soil. So that is how we could get this much difference. But our soils have native endophytic mycorrhizal fungi. So what will happen if we do these studies in unsterile natural soil? So the next few experiments we did, and these are the results. You can see the results in groundnut. This was my first PhD student's thesis work. My name Krishna. As soon as he completed, he was taken as the microbiologist at the International Crops Research Institute at Hyderabad. And, and this is in case of cardamom, this much of difference. And this is in case of neem. Again, why I included these 3? One important, agriculturally important plant, one important in horticulture, one important in forestry. And what is the mechanism by which they improve plant growth? This is the next question. You can see this particular slide, and we just take one example of cotton. And this is uninoculated and this is inoculated. You can see the significant difference in dry weight. And you can see the differences in the phosphorus uptake, zinc uptake, and manganese uptake. So this very clearly shows they help in the nutrition. So here we have to say something about soil science. Nutrients move in soil. Some of the nutrients move very freely in soil solution, and some of the nutrients do not move freely in soil solution. We call them as immobile nutrients or diffusion limited nutrients. So phosphorus is an immobile nutrient, whereas nitrogen, potassium are all mobile nutrients. So let us take of a situation here. This is the plant root, and you have the root hairs. And all of us know that the plants take nutrients from soil through root hairs. So whatever be the phosphorus present in the root hair region, they have taken all the phosphorus. There is no more phosphorus left. But little away, there is plenty of phosphorus, but it is immobile. It cannot come to the root hair. So it is here the mycorrhizal fungi play the most important role. The hyphae of the mycorrhizal fungi, they travel a much longer distance compared to the root hairs, scavenge a larger volume of soil, take the phosphorus and pump it directly into the root system, thereby short-circuiting of the nutrient. This is the most important mechanism by which the mycorrhizal fungi help plant growth. This is true not only for phosphorus, but other immobile nutrients like zinc, copper, etc. Being a microbiology lab, we wanted to study the interaction between the mycorrhizal fungus and the nitrogen-fixing bacterium, Rhizobium japonicum in soybean, and this was a field experiment. Again, I have to say that very few studies have been done under field conditions with mycorrhiza research. So this was one of the very early field experiments done. So there was a synergistic interaction between the two organisms. What I mean by the synergistic interaction, the mycorrhizal fungus increased the nodulation and nitrogen fixation by the rhizobium, and rhizobium in turn enhanced colonization and pea uptake by the mycorrhizal fungus. Thereby, when we inoculate with both the organisms, there was better crop growth as well as the nutrition, and also which was reflected in the grain yield. Dually inoculated plants gave much higher grain yield compared to single inoculation, either with rhizobium or with mycorrhiza. And this was true even for the free-living nitrogen-fixing bacterium, Azotobacter. And again, a synergistic interaction was found. And then coming to phosphate-solubilizing organism, there was a controversy amongst the mycorrhiza workers. Some felt that the mycorrhizal fungi can solubilize insoluble form of phosphorus, because in soil, the phosphorus occurs in inorganic phosphorus, soluble phosphorus, and inorganic insoluble phosphorus. So most of the tropical soils are deficient in phosphorus. So our soils are deficient in phosphorus. Any soil having less than 10 ppm of available P is considered to be deficient in phosphorus. And if you want to know how much is the phosphorus present in Karnataka soils, it is around 2.4 ppm. Where is 10 and where is 2.4? It's so low. Tamil Nadu, it is 3.36. So our soils are very deficient in phosphorus. In addition to that, when a farmer adds phosphatic fertilizer like single superphosphate, 75% of it is converted to a form which is not available for plant growth. So this process is called P fixation. In acidic soils, it is converted to iron and aluminum phosphate, which the plant roots cannot take. In case of neutral to alkaline or saline soils, it is converted to calcium phosphate, which the plant roots cannot take. Plant roots take only phosphorus in the form of H2PO4 ions. So what some mycorrhiza workers believed, that the mycorrhizal fungi can act on the insoluble form of phosphorus through production of organic acids or some other mechanism and release the H2PO4 ion, which the plant roots can take. But another 50% of the people did not believe. So we did this experiment using P32 labeled phosphorus and then proved convincingly that these fungi do not solubilize unavailable form of phosphorus, and they only help in uptake of available P. This can be explained better. When a farmer adds unavailable form of phosphorus like rock phosphate, the phosphate solubilizing organisms act on the rock phosphate, release the phosphate ion, which is taken by the mycorrhizal fungi. So again, there is a synergistic interaction between the two organisms. So dual inoculation is much better compared to single inoculation. And another aspect which we looked into was the possibility of biological control. So you can see this is groundnut plant colonized by the pathogens Chlorotium ralphsei, which is a serious pathogen on groundnut. You can see how sickly the plant looks. But in the next third one, we have introduced the same amount of pathogen plus the mycorrhizal fungus. You can see the difference between the two plants, which very clearly shows the mycorrhizal fungi helps to alleviate the severity of the disease caused by the pathogen. So what's the mechanism? We have worked out, and we found that the mycorrhizal roots have a higher concentration of OD phenols, orthodihydroxyphenol, which prevented the germination of the pathogen. And, and again, another problem in and around Bangalore is the nematode. So this is a tomato root, especially in solanaceous crops like tomato and so on. So tomato root, and you can see the big gall here, tertiary gall. And here we have introduced the same amount of nematodes, nematode larvae, and the mycorrhizal fungus. You can see very small primary gall. So we were wondering as to how to interpret this data. Finally, having discussion with the nematologists, we found that we could conclude that the mycorrhizal fungi do not prevent the entry of the nematode larva into the root system. That is the reason why it forms the primary galls. But it prevents the development of the nematode larva into adult. That's the reason why we are not getting secondary and tertiary galls. And why this is so? This is because we found it is because of 2 amino acids, and which prevented the progress of the larva into an adult. So this was very clearly proved. And what are the other mechanisms by which, or in this particular slide, I have summed up how the mycorrhizal fungi help plant growth. They help the nutrient uptake, especially the diffusion limited or immobile nutrients. They have synergistic beneficial interaction with other soil organisms like nitrogen fixers, phosphate solubilizers, PGPR, and so on. They also protect the plants against the soil-borne plant pathogens. And again, very recent studies have shown that they can protect the plants against drought situation under water stress conditions. And they also produce growth-promoting substances like indolactic acid, gibberellins, cytokinins, thereby promoting plant growth. So it's a cumulative effect of all these which makes the plants to grow better when they are colonized by mycorrhizal fungi. Here I just included one slide to show that this is work done by one of the students. And this is Lucina leucocephala plant. And the first one is the control plant where it was not inoculated with the mycorrhizal fungi. And here they have been inoculated with different mycorrhizal fungi. Why I have included this is what has been said by Dr. Barbara Mose and Professor Gerdiman, the 2 top scientists working on mycorrhiza in the world at that time. They have said mycorrhizal fungi are not host specific. What do they mean by this? When you take the mycorrhizal fungus from wheat plant, try to inoculate banana, it will colonize banana. And you take it from banana and try to inoculate neem tree, it will colonize neem tree. That is what they meant by it is not host specific. Even today, it is true. It's a very correct statement. They are not host specific. But what conclusion we could draw from this experiment study is that though they are not host specific, for a particular host, there is always the best fungal partner where we get the maximum symbiotic response. So this is the best fungus for inoculating Lucina leucocephala. So we introduced a new hypothesis called as host preference in mycorrhizal fungi. So what is the advantage of this? If you are trying to work on tomato and you want to select the best mycorrhizal fungus for inoculating tomato, we can screen different mycorrhizal fungi and select the one which is best for inoculating tomato. So this hypothesis of host preference we proposed, but both the American scientists and the UK scientists did not agree with us. So we have to show it in many other crop plants. This is in cashew, You can see the last one is the best mycorrhizal fungus for inoculating cashew. This is in case of citrus seedlings. The one here, the third one, is the best mycorrhizal fungus for inoculating citrus. With all these, the American scientists said, the Department of Agriculture, United States Department of Agriculture said, if your hypothesis of host preference is correct, we are going to give you a project for 5 years. At the end, we want the best mycorrhizal fungus for inoculating Lucina leucocephala. So the project came, money came in dollars, the university was happy, I was happy, which could help many students research. And what came out of this result? So you can see we selected the best mycorrhizal fungus for inoculating Lucina leucocephala. And there are 4 different varieties of Lucina leucocephala grown in Karnataka. So we selected the fungus which can improve the growth of all the 4 cultivars. The reason is, if one farmer or forest nurseryman is growing one particular cultivar, we can't give one culture to him. Another person is growing another variety, we can't give a different culture to him. So we wanted to give one culture which is effective in all the varieties of Lucina. So that happened to be Blomus mossiae. And this particular fungus was released to the forest department. So now people from all over the world accept the hypothesis of host preference in amphifungi. And they always say, we want the best fungus for inoculating a particular crop. The same experiment, the difference in case of root system of Lucina leucocephala and 4 different varieties of Lucina. And you see, in case of agricultural universities, we have to meet the farmers also. So which we have a Krishi Mela once in a year, when the farmers come and the scientists and professors have to tell what has been done recently. So in one of the farmers meet, I told for this the best Blomus mossiae is the best fungus for inoculating Lucina leucocephala. One farmer stood up and said, which is the next best fungus? And I told him, Gycospora margarita, which is named here, is the next best fungus. Again, he came with one more question. What will happen if I mix both the cultures and then inoculate? I told him, I do not have an answer to give you now. Please come next year. I will do this experiment and give you the answer. And we mixed both, and then there was no antagonistic effect on each other, and they proved very well. So we could give the answer that mixing 2 or 3 cultures has no adverse effect. And at this time, the Director General of ICFRE, that is Indian Council of Forestry Research and Education, from Dehradun, visited the university. And the Vice Chancellor brought the person to a microbiology department, and he brought him also to the glass house. I showed him the difference in case of Lucina leucocephala with and without mycorrhizal inoculation. And he was surprised and said, can your tiny little fungus bring out this much difference in plant growth? I said, sir, what you are seeing is you have to believe. Then he said, okay, we are going to give you a big project. We want the best fungus for inoculating various forestry species. And so these are the various forestry species for which we developed the best mycorrhizal fungus, and it was given to the forest department. And when seeing this, the horticulture department people came and said, we are giving you a project. We want the best mycorrhizal fungus for inoculating various horticultural plants. So we have done and given. I have just included a few of them, not all of them, to be given to the horticulture department. Then the National Medicinal Plants Board came to us. We want the best fungus for inoculating various medicinal plants. So we have developed for the various medicinal and aromatic plants which are listed here. In addition to that, there are many more also, which we have given to the Medicinal Plants Board. Okay, after selecting the best mycorrhizal fungus, what next was the question. How will it be helpful to the farming community, the end user? So here in the x-axis, you can see the increasing level of phosphatic fertilizer. In the y-axis, we have the plant biomass. Okay, now this is the non-mycorrhizal host plant, and that's a mycorrhizal plant. So for the non-mycorrhizal plant to produce that amount of biomass, how much of phosphatic fertilizer it requires? Let us say 70 kg. For the mycorrhizal plant to produce the same amount of biomass, how much of phosphatic fertilizer it requires? Let us say it is 50 kg. That means we are saving 20 kg of phosphatic fertilizer through inoculation with the mycorrhizal fungus. So this caught the attention of many of the funding agencies in Government of India, because we are importing the raw material for manufacture of phosphatic fertilizer in our country. The raw material comes from Morocco. So that means we'll be saving quite a bit of foreign exchange if we can avoid this import or lessen this import. And this we have done in many crop plants. For I have just included one in chili. The recommended level of phosphatic fertilizer for chili is 75 kg per hectare. And the uninoculated plant with the gives how much? 1.81. That is recommended level of phosphate with the recommended level of phosphatic fertilizer. And here we have reduced 50% of the phosphatic fertilizer, and we have inoculated with various mycorrhizal fungi. Let us see the last one. See the amount of yield. Okay, and that means we can save very clearly 50% of phosphatic fertilizer through inoculation with a specific mycorrhizal fungus. So this really caught the attention of the various funding agencies and was introduced as a thrust area of research by Government of India. And nowadays, we all talk of high-tech agriculture. Okay, in earlier days, the farmer himself used to raise the nursery seedlings and then plant it in the field. Now, at least in and around Bangalore, no farmer raises the seedlings. He goes to a nursery man, buys the seedlings and plant it in the field. So this is how the nursery man raise the seedlings in root trainers. And this is the inner view of the root trainer, and where it's very easy to add the mycorrhizal inoculum, raise the seedlings. The farmer buys the seedlings and plant it in the field. So the mycorrhizal roots are taken to the field straight away, and it works extremely well. And at this time, the horticulture department came again to us and said there is a problem in citrus nurseries. The problem is it takes 24 months in the nursery for the root stalks to stay, because they should reach the stem should reach the pencil thickness when they are ready for grafting. And it takes 24 months sitting in the nursery. They came and asked whether the mycorrhizal fungi can help. So we tried various mycorrhizal fungi, and you can see the last mycorrhizal fungus. And this is uninoculated control, and you can see the difference in stem girth. So they were ready for grafting in 18 months instead of 24 months, which was the citriculture nurserymen were so happy because they can save 6 months of the seedlings in the nursery. And this is a routine practice which is followed by citriculturists now. At this time, the floriculturists came to us. We know that in and around Bangalore, floriculture is a big industry. I have to use the word industry because they grow a lot of flowers in poly houses and it is exported outside the country. So most of it goes to Amsterdam, and the flights carry full of flowers. And this I have just given you. Those people came and said how mycorrhiza can help us. So you can see here I included only one slide of chrysanthemum, uninoculated versus inoculated, increased number of flowers. But more than that, what was interesting is the inoculated plants started flowering 3 weeks early. They were more interested in that, in addition to the increased number of flowers, because they can take more number of crops though it is 3 weeks, probably they were more interested in that. At this time, the post-harvest technology professor came to me and said, can I put these flowers in the flower vase, and see whether they stay longer? I said, ma'am, why don't you take and try? And she came back and said, your mycorrhizal plants, flowers stayed 3 days more in the flower vase compared to non-mycorrhizal. Now, if we see, there are so many reports from other countries that it increases the vase life. Mycorrhizal flowers have a more longer vase life compared to non-mycorrhizal flowers. Again, the Indian Council of Agricultural Research came to us with another problem. And what they said is, when the farmer raises cashew seedlings in poly bags and then plant it in the field, there is 40% mortality. That means when he plants 100 seedlings, 40 of them die. That is because of transplant shock. And so they said whether the mycorrhizal fungus can help us. So we did this experiment. The whole thing was done in a place called Ullal, where we have the cashew research station. And the result, you can see, when we raised the seedlings with the mycorrhizal inoculation and they were planted in the field, when they were inoculated with these, the first 2 mycorrhizal fungi, Acylospora laevis and Glomus etericatum, the survival was 100%. Whereas the uninoculated ones, it gives the survival was 63%. Again, the thing which is commonly followed by the cashew growers now. And at this time, the tissue culture people came to us and said how mycorrhizal fungi can help in tissue culture plants. So this is what we did. Introduce the mycorrhizal fungus at the time of hardening. So you can see the difference between the uninoculated and inoculated. It works very well in the micro-propagated plants. And this is another slide with the micro-propagated plant. The first one is inoculated with the plant is Ficus benjamina. This is a plant which is planted in Bangalore very commonly if there is a double road in between, and because they can tolerate pollution. That is the reason why this was selected. And you can see that this Ficus benjamina, micro-propagated ficus, was inoculated with only mycorrhizal fungus. And the last pot, you can see inoculated with the microbial consortium, consisting of the mycorrhizal fungus, a mycorrhizal helper bacterium, and a biocontrol agent Trichoderma aureanum, which gave us a clue that inoculation with a consortium is much better in improving plant growth compared to inoculation with mycorrhiza alone. So we concentrated later on developing consortia rather than only mycorrhizal fungus. So uninoculated acacia, inoculated with only mycorrhiza, inoculated with the microbial consortium. And this is again a DPT funded project which we completed about 2 years back. We know that in case of tomato and capsicum, we have a wilt disease complex. Why we call it a wilt disease complex? Because it is not caused by one pathogen. It is caused by 3 pathogens. It is a fungal pathogen, a fusarium, and it is also caused by a bacterial pathogen, Ralstonia solanacearum, and also nematode pathogen, Merodogyne javanica. That is why we call them as wilt disease complex. Very common in tomato and capsicum. And so the project was to develop a microbial consortium which can protect, even in the seedling stage, which can protect them so that healthy seedlings can go to the field. So we developed a microbial consortium consisting of a mycorrhizal fungus, a PGPR, Pseudomonas fluorescens, and a nematode trapping fungus, Bacillomyces liliaceus. You can see the difference between the two. And of course, for this, this is in case of capsicum. This is not in protrace, but it is even in the raised nursery bed. You can see the difference between the two, and the difference in the root system also, with a control and also inoculated with the microbial consortium. And we have taken it and then planted it in the field in a wilt sick soil containing the 3 pathogens. And we could see that it improved the growth, nutrition, and protected the plants against the disease. And another thing we could another crop to which we could develop the consortium was pacholi. Pacholi is a aromatic crop plant. What we mean is it is used for it is cultivated for extracting scent, the perfume from this particular plant. So this is inoculated with the microbial consortium. You can see the difference. And when the student when she was working with this particular plant, the plants after harvest, she used to shade dry it in the laboratory. And not only the microbiology lab, the entire building used to smell so good because of the fragrance of patchouli. So people used to come to my lab and ask, what you are doing in your lab? Such a good fragrance. So that is all because of this particular plant. This particular plant is grown in a particular district in Karnataka state called Uttara Kannada. And there is always a buyback arrangement with the perfume industry. So they are very much interested in using this particular consortium. And the perfume is, the fragrance is because of the oil it contains. You can see uninoculated control, the oil content was only 2.66. But with the consortium consisting of so many organisms, with the 50% reduction in the nitrogen and phosphatic fertilizer, you can see the oil content was 3.66 compared to 2.66. So this is where the industry is very much interested in producing the, industrially producing the perfume. And another example I want to say about the microbial consortium is Coleus fusculi. Is a medicinal plant that was introduced in 3 southern states. It was introduced in Tamil Nadu, Andhra Pradesh, and Karnataka with the buyback arrangement, with the buyback assurance from the pharmaceutical company. So because it contains a particular compound, forskolin, which is used in so many variety of medicines. So the farmers were very happy. They raised it in a large scale. But what happened? There was a serious disease problem, Fusarium wilt. And this is one of the experiments which was done in a place called Kollegal, a field experiment in a farmer's field. And you can see in the control, what was the disease index is 85% in disease. And when we inoculated with only the mycorrhizal fungus, the disease index came down to 68. When we developed the consortium with trichoderma, it came down to 33. So again, showing that microbial consortium is much better than mycorrhizal fungus. Through another project, DBT gave us funding to develop a inoculum, mass production technology, and we selected the best host, best substrate, etc., etc. So this is now taken by 4 industries within the country and 2 industries outside the country for mass production and for the production of mycorrhizal inoculum. The next question was, which is the best place to place the inoculum to get best plant growth response? And in case of raised nursery beds, as the farmers maybe used to do, placing the inoculum 2 centimeters below the seed gives the best results. In poly bags, placing the inoculum 6 centimeters below the seed gives the best result. And how much of inoculum has to be added? In case of plants raised in nurseries, about 1 kg per meter square, but now we have come down, even 500 grams per meter square gives the best result. And here again, a farmer's day came, Krishi Mela, and we have to face the farmers. So one farmer raised and asked, I have a 10-year-old mulberry garden. Will your mycorrhizal inoculation help in improving the leaf yield? Because mulberry leaf is the most important part. Will it increase the leaf yield of the old plant, like 10-year-old plant? Again, I have to say him, I don't have any experiment done on this line. Please come next year. I will give you an answer. Fortunately, the sericulture department in the university had a 10-year-old mulberry garden, And we introduced the mycorrhizal fungus in the root region, and then we could see that the yield increase was there, and it was as good as giving 100% the phosphatic fertilizer. So even in the old trees, we have confirmed this with another perennial crop like clove, and this was done in the Spices Research Institute in Kerala. And for the older plants, it was introduced in the root region, and where we could get increased yield. And the Planning Commission wanted to know about the economics. They invited me as a special invitee to one of the Planning Commission meeting, and we said, don't know biology, we want only economics. So we worked out this particular table for them, and this is, I did it with the tomato crop, which experiments we did with the tomato crop. The yield of tomato crop, uninoculated tomato crop, given 100% of phosphatic fertilizer, the yield is 9,000 kg per acre. When the amount of phosphatic fertilizer was reduced to 50%, inoculated with the best mycorrhizal fungus, the yield was 10,400 kg. That is 1,400 kg increased yield. And the price of tomato in Bangalore changes. It can come to 2 rupees per kg sometimes, it can go to 100 rupees per kg sometimes. I have taken 25 rupees per kg on an average. So that means the farmer will get 35,000 rupees more. And he has saved 50% of phosphatic fertilizer. So totaling 36,875 rupees he will get per acre of tomato grown through inoculation with the best mycorrhizal fungus. So the bureaucrats with all the IAS officers who attended the meeting were very much convinced, and they said this can go as one of the thrust areas of research. And I was to retire at that time from the university, and I was a member of the expert committee of Department of Science and Technology, Government of India. And at that time, a question was asked by one of the DST officials, what you are going to do after retirement? I said, I'll continue my research as emeritus professor in the university. They said, that is fine, you continue as emeritus professor, but there you cannot do 100% concentrate fully on research. So why don't you start a trust and then concentrate on microbiology research? So that is how we started this Center for Natural Biological Resources and Community Development, and started doing research. And we wanted a research project. The rules of Government of India, when a trust is formed, it has to wait for 3 years before a project can be sanctioned. Department of Science and Technology said, we can waive off this rule because you are already a committee member, or expert committee member. So immediately I got this project, but with the condition was what they said, your students have done excellent work, no doubt about it. And how many plants they have used? Maybe 100 inoculated, 100 uninoculated plants, and shown the difference. But suppose a farmer raises thousands of seedlings, will we get same result? So they said the first project, you have to have 3,000 seedlings. The same thing what you have done with your students, but 3,000 seedlings. I was scared because there was only one research fellow granted. How can one research fellow raise and do the inoculation, observe 3,000 seedlings? At this time, the Department of Forestry, State Karnataka Department of Forestry came to my rescue. And what they said, don't worry, we'll give you as many labor as you want. Could be 30, you want 30 or 40 laborers, I'll give you. But what we want is to see how it works in a large scale. So what I did was, we selected 3 tree species. So these are the 3 tree species: Tectona grandis, Dalbergia sissu, and Acacia auriculaformis. You can see difference between uninoculated and inoculated seedlings. So you can even in Dalbergia sissu and Acacia auriculaformis. So I was very happy that what my students have done works even on a large scale. Even when we have 1,000 seedlings of each one, 500 uninoculated, 500 inoculated. So with 3,000 seedlings, we could grow. It works very well. And there is a degraded forest in a place called Mandya. Most of this research was done in a place called Mandya. Mandya is in between Bangalore and Mysore, and there is a degraded forest there. You can see the soil, how degraded it looks. And so we planted these seedlings in the degraded forest. Again, the Karnataka Forest Department gave us plenty of labor and wanted to see how it works. So you can see the difference. This is 1-year-old seedlings, uninoculated versus inoculated. And same thing happened with Acacia auriculae also 1-year-old seedlings. And here, that down, you can see the one which is at the bottom. This is Dalbergia sissu after 52 months. After 52 months, the forest man looks big. This is uninoculated. And this is inoculated Dalbergia sissu, and he becomes small. Why? Because the plants behind him are big. So the inoculated plants were inoculated plants were twice the size of the uninoculated plant. So that at this particular thing, the Department of Science and Technology, which gave the funding for this project, I invited the scientist in charge, Dr. Vinita Sharma. She came to Bangalore. I took her to the forest, and she saw this and said, we should have such replications done all over the country in various states. And I don't know when we are going to do it. I don't have time now to do that. And we also organize training programs for the farmers and also the young scientists who come. We train them in mycorrhizal research, and then they go back and try to do it. So finally, to conclude, what I can say, at the time of independence in 1947, there was shortage of food in our country. So we were very keenly waiting for the ship to come from United States carrying wheat. And that is why we call it as 1947, that period, ship to mouth existence. So when the wheat came from US, then we were very happy to grind it and make chapatis or rotis and then eat. So that was the condition. There was insufficient food. Thanks to Green Revolution, high-yielding varieties of crop plants were introduced, and the food production went up. So there was enough food for people to eat. But after several decades, we know what are the bad effects. What are the bad effects? For raising these high-yielding varieties, we have used heavy doses of fertilizers and pesticides. And now we know these fertilizers and pesticides are deleterious to soil health. When it is deleterious to soil health, it is also in turn deleterious to plant health, animal health, and human health. And the soil in the groundwater, we find the nitrate, recommended amount of nitrate is several times more than the recommended level of toxic limit. And same thing with the pesticides. The tender coconut water, what we are drinking contains pesticides. All these makes us to feel that we should switch over to sustainable agriculture. What we mean by sustainable agriculture? Use less chemicals like fertilizers and pesticides, use more biologicals. So here, the mycorrhizal fungi and other microbial inoculants play a very, very important role. They are cost-effective, much cheaper compared to chemicals, environment friendly, this is very important, and they improve soil health. And now there is a lot of funding available from the global institutions like FAO, World Health Organizations, to improve soil health, which in turn will improve the plant health and in turn animal and human health. So these microbial inoculants, including mycorrhizal fungi, they are a boon to farmers, especially in developing countries like ours. So with this, I conclude and thank you very much. And if you have any questions, you can ask me, or better to put it in the chat box.
Yes, sir. Thank you so much, sir, for your wonderful lecture. We have learned the very basics of the mycorrhiza, sir. and I'm sure it will be much useful for the young minds like graduate students, postgraduate and undergraduate students. And they have also posted some of the questions, sir.
Okay, okay.
One of the students have asked how long the mycorrhizal fungi can survive without the soil or water.
Pardon?
How long the mycorrhizal fungi can survive without the soil or water?
Provide nutrients.
Nutrients, without nutrients.
Oh, no. I don't say that without 100% nutrients. They do require some quantity. That's why we always say 50%.
Yes.
We can reduce it to 50% of the chemical fertilizers.
Yes, sir. And one of the another participant asked, can plants survive without mycorrhiza? Is it essential or mandatory that a plant should have associated with the mycorrhiza?
Okay. The thing is, it's not mandatory except for few plants. Okay. So when there are soils, when they are having less P, then it is mandatory. They have the colonization. In a soil which is very rich in phosphorus, then they don't require the assistance of mycorrhizal fungi. But there are, for example, orchids, I was telling, different groups of mycorrhizal fungi. In case of orchids, it is mandatory they should be colonized by the mycorrhizal fungus. If they, when the seeds of orchids, they imbibe water and they germinate, and we call that swollen orchid seed as protocorm. And they wait for the infection by the mycorrhizal fungi for 6 months. If they are not colonized by the mycorrhizal fungus, they do not develop into a plant. To that extent, they are obligatory dependent on mycorrhizal fungi.
Yes. Yes, sir. Thank you, sir. And one more participant has asked, do mycorrhizal inoculation can be applied for any plant? Can we apply for moringa, for instance?
Yeah. See, there are certain plant families which are not colonized by mycorrhizal fungi. The one which is very common, the plants which are listed are plants belonging to Brassicaceae, which we usually call the cruciferae, Chenopodiaceae, Polygonaceae. These are some of the families where the mycorrhizal fungus, they do not prefer to colonize. We can force them to colonize, but naturally they are not colonized.
Yes, sir. Yes, sir. Thank you, sir. Myself have a couple of questions, sir.
Okay.
So what could be the effect of these mycorrhizal associations or the effect of the pesticides on these mycorrhizal associations, sir? Do the pesticides have any effect?
Pesticides?
Yes, sir. Pesticides, herbicides.
Yeah. Okay. Okay. To answer this question, it's very difficult because there is a very nice reviews on the effect of pesticides on mycorrhizal fungi, and some pesticides or we can say most of the pesticides have adverse effect on mycorrhizal fungi. Some of them have no effect, and some of them stimulate mycorrhizal fungi. So we have 3 categories.
Okay.
I usually, when I talk of ecology, I go into the details and give all these pesticides which promote and all that. Yeah.
Yes. So, can we genetically engineer a mycorrhizal fungi so that it could become a resistant strain against the broad spectrum herbicides or fungicides that we apply?
Broad spectrum?
Fungicides or herbicides. Can we genetically engineer a mycorrhizal fungi so that it acquires resistance against the broad spectrum herbicides and fungicides? We apply.
The working with the genetics of mycorrhizal fungi is very difficult because they are obligate symbions.
Yes,
we do not have the culture in the lab to do all this, so it makes it very difficult. But that's why people have not gone into that area of genetically modifying fungi. Whereas it is possible to do with the ectomycorrhizal fungi, which we can isolate and have the culture. It is true with case of required mycorrhizal fungus also, which can be brought into culture in the laboratory. It's also true with orchid, but with arbuscular mycorrhizal fungus, it is very not possible.
Okay, sir. Yes, sir. Thank you so much, sir. I think there are no more questions they have posted in the YouTube's live stream. Okay. So I once again thank you, sir, wholeheartedly for your excellent deliberation on this technical session 6 of this 2-day Science Academy's lecture workshop on recent trends in microbiology and biotechnology. So I think we can have a break, sir, here, and we'll be joining by 2 o'clock for the technical session 7 by Dr. N. Tajuddin again. So we'll have a break.
Great. Good. So, Professor, it was a nice lecture.
Oh,
I
thank
heard
you.
it all through,
Thank you.
sitting quietly. It was a good one. Yeah, so we meet at 2 o'clock then again.
Yes, sir. Yes, sir.
Thank you. Thank you.
Thank you, sir.
Thank you.
So dear participants, the YouTube link for the session 7 and 8 has been sent to your mail. So please join us in that link in the afternoon session. Thank you.